My name is G.Zubal. I manage the nuclear medicine and X-ray portfolio at NIPIB, as well as grants that are awarded through the BRAIN Initiative. I'll be giving you an overview of funded molecular imaging projects that are active under this effort. Just a quick mention of some NIH resources. If you missed the previous workshop on dissemination of non-invasive technologies, you can see in here an extensive number of talks that cover the funded group grants under the BRAIN Initiative, including the ones I'll be summarizing here. You can do that by clicking on the URL shown at the top of the screen. The 3 grants, as well as other research grants funded by NIH, are available for your perusal at report.nih.gov. This website allows you to search for specific research groups, grant titles, research areas to view their abstracts and additional administrative details. It is really a very good resource to see who is researching on which topics for a better understanding of the research areas supported by NIH. Let me just give you a quick overview of the history for funding opportunities that led to the recently awarded nuclear medicine molecular imaging active grants. The first funding opportunity announcements were issued in 2014, in the early days of the BRAIN Initiative that was brought to Congress by President Obama. And the first full-scale development awards came out in 2019. 3 U01 grants are currently funded from these opportunities, and I'll give you a quick overview of their aims and innovative developments. You see their titles listed here. The first of these 3 active grants is entitled Ultra High Resolution Brain PET Scanner for In Vivo Autoradiography Imaging. It is led by Dr. El Fakhri at MGH at Harvard Medical School. He proposes to build a PET scanner using DOI, or depth of interaction techniques, which localizes the position of interactions, radiation interactions, which improves the spatial resolution of the images. A schematic design of this scanner is shown here on the right. The investigators will implement quantitative methods for in vivo parametric mapping of pharmacokinetics of the acquired human images. And in the bottom right corner, you see a simulation of the expected higher spatial resolution brain images of this proposed camera compared to images currently available on existing cameras. The technology breakthrough here is increased spatial resolution or image quality, benefits to neuroscience, the ability to measure brain dynamics of neuroreceptors in the functioning human brain. The second active grant is headed up by Dr. Katana and is entitled Development of Human Dynamic Neurochemical Connectome Scanner. He is also at MGH at Harvard Medical School, and he proposes to build a very high sensitivity dedicated brain PET scanner in a spherical geometry shown on the right. And it will fit into a 7 Tesla MR scanner. The innovative non-cylindrical design will require the co-development of the reconstruction software necessary for creating the brain images from the camera data, and the detectors used in this proposed camera will also implement depth of interaction methods for improved spatial resolution. The technology breakthroughs, spherical camera design, and benefits to neuroscience combine PET and MR images in the same task or same condition of the subject's brain. The third active grant is entitled Ultra High Performance Human Brain PET Imager for Imaging Neurochemistry, and is supervised by Dr. Carson at Yale University's PET Center. He proposes to use a PET camera using the collaborative efforts of United Imaging Healthcare and the PET group at UC Davis. This design will build upon the successes of the first whole body PET camera at UC Davis and will implement a 50 centimeter long cylinder surrounding the subject's head, as shown in the top right image. An additional technology breakthrough will employ a real-time marker-free head motion correction system, shown here aligning two positions of the same subject's facial features, one shown in red and the other in green. This alignment will remove artifacts due to the patient motion during the camera acquisitions. The neuroscience breakthrough will allow imaging of small brain structures, raphe nuclei, substantia nigra, to capture their neurotransmitter dynamics. As you can see, these three PET camera development grants are similar in that each proposes to engineer a next generation PET camera with improved spatial resolution and higher sensitivity for faster imaging times. Each investigator approaches these improvements with their own innovative ideas, and all have the combined goal of imaging smaller brain structures and producing dynamic sequences of neurochemicals that underpin the human brain function. We believe that each of these approaches will bring unique contributions to obtaining improved images of smaller structures, leading to new insights about the functioning of the human brain. Thank you for your attention.